Hello. First of all, I want to acknowledge the invitation from Andy and from Norberto to present this study in this very interesting symposium. As you know, and we have been talking about that, many patients with epilepsy present mood disorders. There are many hypotheses to understand why these patients present both epilepsy and mood disorders. One hypothesis says that both epilepsy and depression and anxiety are manifestations of changes in specific neurotransmitters. Other hypotheses established that epilepsy induces important changes in the brain that facilitates the depression and anxiety. However, and unfortunately, both depression, mood disorders, and epilepsy involves many, many mechanisms. In this talk, I'm going to talk about different receptors changes that could explain the comorbidity of epilepsy and mood disorders. We have to understand that the mood is regulated by several neurotransmitters. For example, serotonin, norepinephrine, and dopamine. Concerning dopamine, it is very well known that it acts in different receptors. Some of them induces excitatory effects, other induces inhibitory effects. The receptor with the higher affinity to serotonin is the 5-HT1A. Therefore, many studies have been focused to understand what is going on about this receptor and epilepsy and mood disorders. Concerning depression, it has been described that there is an important reduction of receptor binding in specific brain areas such as RAFE and mesial cortical structures in patients with depression. Similarly, in epilepsy, many studies identified a significant decrease in the 5-HT1A receptor in different brain areas, but most important in the areas that correspond to the hippocampus. Here you can see one of the sections that corresponds to a control. In yellow, you can observe the high affinity of 5-HT1A receptor. The other slices correspond to patients with mesial temporal lobe epilepsy, and you can see there is a significant reduction of receptor binding in the temporal lobe ipsilateral to the epileptic focus. We could reproduce these changes using in vitro autoradiography. In this study, we evaluated different receptors for serotonin and also the transporter. What we found was that there is an important reduction in 5-HT1, especially in the layers 1 and 2, and also there is an important reduction in the binding for the transporter to serotonin. And the last year, Theodore and collaborators described for the first time that the reduction of receptor binding 5-HT1A corresponds to a high rate of anxiety and depression. In other words, patients with these disturbances and epilepsy present very low levels of 5-HT1A receptor. In summary, we can see that epilepsy and mood disorders share this kind of alteration, a significant reduction of 5-HT1A receptor. And most interesting is that many substances used as an agonist of 5-HTA receptor and used for reduce the depression are also inducing anti-epileptic effects. Other system is the dopaminergic system. This system is involved in different functions such as the reward, the pleasure, motor function, compulsion, and perseveration. It's involved in functions like the appetite, and also in some physiological functions like the sensation of pleasure, the feeling of pleasure. It is described using PET studies that when a person is listening a nice music, in her brain there is a very important activation of the dopaminergic system. Also, during the process of falling in love, the person presents a very, very important activation of the dopaminergic system. Unfortunately, this situation disappears more or less 4 years later. And also, dopaminergic system is very active when a person is addicted to some specific drugs. Unfortunately, this leads to addiction. How about epilepsy? Well, we have to start saying that there are many types of receptors for serotonin. Some of them induces inhibitory effects, dopamine receptors. Some of them induces inhibitory effects. I'm going to talk about the D2 receptor. Concerning anxiety, there are evidences indicating that the D2 receptor binding is decreased in the brain of patients with high rate of anxiety. About depression, the results are confounding. Some studies, like this one, indicate that patients with depression present no significant changes when compared with the control. However, other studies indicate that D2 receptor binding is enhanced or decreased in persons with depression. How about in epilepsy? In epilepsy, there are only 2 studies. One of them was carried out by Berham, and he did some PET studies and found that in temporal neocortex, ipsilateral to the epileptic focus, there is a significant decrease of the D2 receptor binding. We couldn't replicate this finding using in vitro autoradiography. As you can see here, there are no significant changes in the different cortical layers of patients with mesial temporal lobe epilepsy. However, it is important to notice the high variability of the values. All of these experiments indicate that receptor binding, D2 receptor binding, could be involved in both mood disorders and epilepsy. However, it is important to remember that these strategies only label the receptors and they don't give any information about the transcriptional mechanisms. Therefore, we were interested in to analyze the capacity of the D2 receptor to activate the G protein and then the signal cascade. What we did was to label the G protein activated by an agonist of the dopaminergic D2 receptor. What we found was, first of all, the patients with epilepsy and anxiety and depression present no significant changes when compared with the control that were autopsies. However, it was very interesting to observe that those patients with epilepsy and without mood disorders present an important activation of the G protein induced by D2 receptor binding. Then all these experiments give the information that in some way dopamine is involved in both processes, in both disturbances. There is the general idea that both present a decrease of the dopaminergic neurotransmission. However, our studies indicate that patients with epilepsy without mood disorders present a very important activation of the G protein. That means that in some way we have to evaluate also these patients that do not present mood disorders, because probably that is the key that we have to look for the reason and probably apply it in order to avoid the comorbidity. GABA system. GABA system, as you know, is one of the most important inhibitory neurotransmitters in the brain, and it is involved in both mood disorders as well as in epilepsy. Concerning depression, and using PET studies, it has been described that the benzodiazepine binding is significantly reduced in patients with depression. In epilepsy, there are some studies using in vitro autoradiography, and you can see here that patients with epilepsy present no significant changes when compared with the control tissue. How about benzodiazepine receptor binding and epilepsy? There are several studies, some of them indicating an important enhancement of benzodiazepine receptor binding. However, others indicate that there are not significant alterations. Therefore, in general, the idea is that in depression, there is a decrease of the GABAergic neurotransmission, whereas in epilepsy, the results are conflicting. But what could be the reason? The reason could be the analysis and the strategy to evaluate these kind of receptors. For example, in vivo tomography, it is impossible to avoid the endogenous ligand, and therefore the ligand, the external ligand, is competing with the endogenous ligand. The other strategy is is autoradiography. Therefore, it is possible that these conflicting results are due to the different strategies. Therefore, we decide to analyze in some way what changes present patients with epilepsy and mood disorders and patients with epilepsy without mood disorders. This is a polar plot graphic in which we evaluate GABA-A and benzodiazepine binding. In the A side, you can see in this side, you can see the different values obtained from patients with epilepsy and depression and anxiety in general. In contrast with the control group marked with blue, it is possible to observe that there are not significant changes for GABA-A and benzodiazepine receptor binding in red. But what is going on with those patients? With those patients with epilepsy and without mood disorders, we observe as increase of benzodiazepine receptor binding in all the cortical layers of the cortex, temporal cortex. This effect or this alteration is also associated with a significant decrease of the GABA tissue content in temporal lobe epilepsy in patients with epilepsy and mood disorders. In some way, it is easy to understand that although these patients present no significant changes in GABA-A and benzodiazepine receptor binding, they don't have enough GABA to stimulate them. And this situation is very similar to that observed in patients with Alzheimer's disease and depression. There is a significant decrease of GABA. In general, we can conclude that mood disorders is associated with a significant reduction in the neurotransmission induced by GABA. How about in epilepsy? In epilepsy, we can suggest a significant decrease in the neurotransmission induced by GABA. However, why the benzodiazepine receptor binding what are they doing in the brain of patients with epilepsy without mood disorders? It is very well known when the benzodiazepine receptor binding is active, it facilitates the GABAergic neurotransmission. Then, if those patients present higher benzodiazepine receptor binding, obviously, they present an enhanced GABAergic neurotransmission. Then again, I think it's very important to analyze the brain of those patients without mood disorders in order to understand the mechanisms they present in order to avoid that disturbances. However, at this moment, there are many studies indicating and evaluating the receptor binding, but probably the key of these disturbances is in the transductional mechanisms downstream the activation of each receptor. Therefore, we look for information about changes at that level in patients with depression, and the expression of the G protein present different changes depending on the area, depending on the model, but in general, there is an agreement that the adenylatosclasa cascade is decreased in these patients. How about in epilepsy? In epilepsy, also, there are confounding results about the G protein expression. However, the CAMP and the adenylatosclasa expression are enhanced. In this case, we analyze the expression of the different proteins of the GE alpha subunit. What we found was that patients with depression and anxiety, as well as patients without mood disorders, present no significant changes in the different expression for the G protein. However, for patients with mood disorders, the GABAergic, the adenylatosclasa binding is increased, but this effect is much more evident for those patients without mood disorders. In some way, these results indicate that there is an overactivation of these mechanisms that probably are involved in avoiding the mood disorders. Well, at this point, I have been talking about the receptor binding and their signal transduction mechanisms. It is clear that many drugs are focused to activate specific receptors in order to have specific effects. However, since 2008, Fuchse has been described that many receptors coupled to G proteins can interact between each other and can form homodimers or heterodimers. The functional consequence of this situation is that the mechanisms, the cascade of mechanisms of the signal transduction are completely different to those observed for each receptor by itself. How about depression? This study was very interesting. Pei and Louis in 2010 described for the first time the presence of heterodimers in the brain of patients with depression. Then they used an experimental model of depression and applied a peptide that interact and block the action and the effects of this heterodimer, and they found that the animals present a decrease of the depression behavior. Therefore, in the future, I think we have to think about these new targets in order to treat depression or the comorbidity of epilepsy and depression. Finally, I'm showing here the god of the epilepsy for Mexico, the goddess Tlazolteotl, and this is the people who has been collaborating in our studies. Thank you.